Hi guys, welcome to Biology Lesson 3.08, the second part of active transport or active transportation. So in our overview, we talked about the fact that we had two different categories. We've got active transport where the whole cell membrane is involved, and we have active transport that is a protein, which would be the sodium-potassium protein, sodium-potassium pump protein. So we're going to start off talking about endocytosis and exocytosis, and this involves the whole cell membrane. And we had said we have a phagocytosis. Phago translates to eat, and pino to drink. Cyto is cell, so we said it was cell eating and cell drinking. And I know this is macromolecule. We're going to go with a pretty large structure. Here we would have smaller molecules or liquids. So, but both of these are examples of endocytosis. Endo is into, and then cyto is cell. So these are how we're taking these things into the cell. So we start off, and we have something that's approaching the cell. Now, the cell doesn't automatically take everything in. You have to have the right receptor. You have to trigger the right button. And remember, we talked about viruses doing that. Viruses come up and they dock with a receptor, and the cell goes, oh yeah, that's something I needed, and they bring it in. So viruses actually trick the cell. But in this case, it's something the cell wants. And so what will happen is the membrane will start to fold a little bit, and then the divot gets a little bigger, and it starts to kind of fold in around this particle, this very, very large piece of whatever it is. And it folds and it begins to pinch together, and the sides are moving together like this, and eventually they come all the way together. They pinch, the cell membrane's closed, and you end up with your particle inside of the cell like this. Now, this blue circle that used to be part of the cell membrane is called a vesicle. A vesicle is just a circular membrane, or actually it would be spherical. This is 2D. A spherical membrane. Can't spell today. That carries stuff around. So we use vesicles to transport stuff. And we have the same thing happen over here, except with smaller molecules. But we end up with this vesicle with the stuff inside of it. What if we want to send something out of the cell, exocytosis? We start with a vesicle. So here I have a vesicle, and maybe it came off at the end of one of my organelles, and it's got something I want to send out of the cell. What happens is this vesicle comes up and it fuses with the cell membrane. And then slowly this begins to open, and until it's completely open to the outside. This is outside the cell, and this is inside the cell. And we release whatever it was we were trying to release. And now my vesicle becomes part of the cell membrane. So with exocytosis, we go from vesicle to cell membrane. And with endocytosis, we go from cell membrane or plasma membrane to vesicle. But this takes a lot of energy, and it takes it in the form of ATP, because we're pulling in a large portion of the cell membrane. This movement doesn't just automatically happen. It requires power. And the analogy we gave was, if we were a car dealership, the gases go in and out around the windows. You don't have to worry about oxygen, carbon dioxide. People can come in and out through the doors. But if you're going to take a car in and out of the showroom, you've got to open up an entire wall, and that's what this is equivalent to. So we're taking things in by big changes in the cell membrane. Right, we're going to flip back to this page, and I'm going to add another page. I'm going to hit the pause button. Okay, we've already done this side of the sheet. So this is our active transport, and what we have here is our sodium-potassium pump. And it's the protein. So we could call it the sodium-potassium pump protein if we wanted to. Protein. So it's this guy right here. This is the sodium potassium pump protein. And if you look at a diagram, maybe at the end of this video, I'll pull up the actual diagram of the sodium potassium pump protein. But this is outside the cell. This is inside the cell. This is my phospholipid bilayer. So this is going to be an integral protein. We talked about carrier proteins. We talked about ion channels. This would be, I suppose, in the category of a pump protein. But in this case, all of these little circles are sodium. And these guys are potassium. Well, based on everything we've learned so far, what direction should the sodium go? I have one sodium inside the cell and I have 5 outside. So what we've learned so far is that the sodium should go this way, but that's not what happens. The sodium potassium it's going to pump more sodium this way, and it's going to bring potassium in. And so it's actually going to make this worse. We're going to keep adding sodium outside the cell. And we're going to, instead of releasing the concentration, letting the concentration equalize, we're going to make it worse. We're going to make sodium more concentrated. So this actively concentrates, actively creates concentration. I'm going to abbreviate it. Well, why do we do that? What happens is the cell, when the cell needs it, all of a sudden it'll open up a gate here in the membrane, and all of that sodium will rush through the gate. Boom, 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 boom. And we'll do work with that. We'll do chemical work. But to create this concentration takes a lot of ATP. Not just a lot. 60% of all the ATP, 60% of all the ATP your body makes gets used to create this concentration of sodium. The sodium potassium pump protein takes over half of your ATP. That's how important it is to build up these sodium concentrations. Well, where do we use this? Every nerve cell that you have. Every nerve cell requires this concentration of sodium. Over here, to compare it to, we saw that all of this goes from high concentration to low. When we go from low concentration to higher concentration, we're going in the opposite direction. This is called going with the concentration gradient. So passive transport, we say, goes with the concentration gradient. But this active transport sodium potassium pump goes against the concentration gradient. Because we don't go from high to low. We go from a lower concentration to higher. We make it more concentrated. Okay, that kind of is our summary. So we talked about cell membrane active transport, endocytosis, bringing things in, creating vesicles, exocytosis, vesicles into the cell membrane. And then we wrapped up with this active transport, creating a situation where we have more concentration that we can use later. And the analogy we gave in the last video was putting water up into a water tower. And then when you need a shower, the water has, gravity can pull it down. And this way, the concentration gradient will pull this in when we need to do work with it. All right, that's all we've got for active transport. And that wraps up this chapter. Thanks for watching and have a good day.